Oh my god, yesterday was so stressful. I spent literally the entire stream doing nothing but a single question on the Hardy-Weinberg principle, a 2-mark question. And I was doing all sorts of math. I was putting all sorts of formulas in a calculator, trying to figure it out. I couldn't get anywhere. I watched a 1-minute YouTube video. I have it cracked. I have it all on lock now. So let me quickly explain what I figured out before we move on to. I want to do some actual revision today. This is like the easiest biology 2 marker right here. I want to do some actual revision on the sliding filament theory, and then we'll crack on with paper 2 revision, because we're about halfway through paper 2 now. So yeah, let's get on with this. So let me quickly explain. So this is obviously the Hardy-Weinberg equation. Now the thing that I was not understanding is what 0.36 was. So we were given, biology is just like, it's just an evil version of math. What we were told is that 36% of the, I guess it helps to have a reference number. Let's say that out of 100 organisms, 36% have the gene P, okay? That means that P cannot be equivalent to 36%. This entire thing is 36%, because it's the allele. That's the bit that I got wrong. I assumed this was the gene, it's the allele, like the phenotype. I'm saying this out loud and it's very, it's not making sense, but it's making sense in my head. Basically, that should how I'm gonna figure out how to draw this. Okay. If we assume that, I mean, I've kind of written out here, capital F, that gives you a white organism. Capital F, lowercase f, also white, because it's a dominant gene, and lowercase f gives you grey, then it means that this is equivalent to 0.36, not one of them. So that is equivalent to 0.64, and all you need to do is square root 0.64. That is literally the only thing you need to do to figure out the frequency of the organism having at least one F allele. And that is so bad, because that question I spent 40 minutes on. I did the square root of P squared, I did the square root of these numbers. It's so easy, but it's actually a very difficult concept to wrap your head around. That's why I wasn't able to figure it out. And that's why I'm struggling to explain it now, because it's a very difficult concept to wrap your head around. What are you doing, actually? What are you doing? Because as you can tell, biology is very fun. That's why I'm doing it in university. But what are you doing? Oh, just a sec. Okay, double spot bit. I can give you some advice for core math. It is blessed. Like, it's very easy. What are like your predicted grades for math? Because genuinely, like, it's easy. It's very, very good. For core math, literally like the first month, I would say it was equivalent to like year 7 math in terms of the things we did. I think you definitely be able to do well at core math. Like, as proof of concept, the first month, we literally did, like, bid mass, I think rounding and, like, things like that. Very easy stuff. It did get up in difficulty, but it's a very common sense subject, core math, where if you literally just have common sense, you could probably get half the questions without revising. Because that's what I did. I didn't revise for the exam, and I ended up doing quite well. I'm just trying to go on the AQA website. But genuinely though, good luck with year 12 and 13, because it's gone by very quickly. Where is the exam materials? Oh my god. Why can't I find it? Am I going... Paper 2? I found it. Yeah, it's very much common sense. Like, some of the questions are rarely common sense. I think it's something like... I think, like, an eighth of a paper is just a single question about, like, estimating something. So literally, you could get a lot of the marks just for common sense, which is good. Oh, this is taking so long to pull up. There we go. Okay. So, last time, we just finished question 4. Oh. Oh, wait, no, I've done this exam question before. Have I? No, this is a different one. Use figure 3 and your knowledge of colour vision to explain how an orange colour is seen at 600 nanometers. Oh, God. Oh. Okay. What are we saying here, then? How an orange colour is seen at 600. I mean, that's just going to be because... How an orange... Wait, have we even been taught this? The absorption of red is 75%. The absorption of green is like 30% and blue is 0. The only thing I'm worried about is... It might be... Yeah, but it's only a year core math. Like, you could really put it on the back, like the backlog, and still do quite well at it, I think. Like, even if you got like a C or a B in it, like, it's still like 10 or whatever 12 or 16 UCAS points. Like, I don't think it's a topic you need to revise a lot. I would argue the homework is arguably the most, like, annoying part of it, just because, like, she gives you a lot of past papers. Right, let us see. This is cone cells and rod cells. It's been a couple of weeks since I've revised this, so I might be a bit rusty, but cone cells have a high visual acuity. Rod cells have a low because of retinal convergence. Cone cells are abundant in the fovea. Cone cells allow you to see a high color spectrum, low light intensity, high light intensity. Is there anything else we need to know? I don't know. Right. You get... oh, okay, yeah. No, I feel like you do quite well at core math, but really, it should come kind of... If you're doing GCSE statistics, I would say, like, a good 2/3 of a course you'd already know, which is really helpful, but even if you don't, like... It'll come very easily, I think. Anyways, use your knowledge of colour vision to explain how an orange colour is seen. I don't understand what this is asking here. Because I just wrote all of this information down, and I'm kind of wondering why I would need to use any of this anyways. Use your knowledge of colour vision. We haven't looked at colour vision much. It's showing that red is absorbed quite a lot, green is absorbed somewhat, and blue isn't absorbed at all. I'm gonna have to look at the mark scheme here. I don't know what it's... Oh, okay, you're taking the piss. 1 mark for literally saying it involves cones. Yeah, okay. Right. Stating the absolute obvious, that's so sad. No. Each type of photoreceptor has a different pigment. We haven't learned that, right? We know about rhodopsin and iodopsin, but what does it mean by a different pigment? That is such a weird mark point. Oh, and you could have just got a mark for stating percentages. Okay, you know what then? I feel like in an exam, I feel like I would have probably put that, I think. Out of 3 marks, yeah, you could easily say cone cells and then just put some percentage values if you had no idea. I think that that's fair then. I don't think that's a bad question. Just, we haven't covered much of that, so... Oh, which of the following statements is associated with high sensitivity to light in the retina? High sensitivity to light, so it's going to be a single photoreceptor connecting to 1 neuron. Oh, temporal or spatial summation. We haven't done this in relation to photoreceptors, though. Obviously, for cone cells, it is literally 1 cone cell connected to a bipolar cell, which then... a bipolar neuron, sorry. Meanwhile, with rods, it's multiple rods connected to the same bipolar. I'm going to assume, then, that it's a single photoreceptor and it's going to be temporal. An increase of frequencies. Wait, no. Fuck, yeah, I knew that. I misread the question. High light sensitivity. Obviously that's gonna be the rod cell, not the cone cell. Oh, okay. I misread the question. Yeah, several photoreceptors, and it's going to be spatial summation, because that's how 1 rod connects to... multiple rods connect to the same bipolar. Okay, next question. We have not done that yet. Oh. Maybe this one? A decrease in blood pressure stimulates the release of ADH. Give the location of receptors that detect a decrease in blood pressure. No, have we done this? Would it be baroreceptors in the carotid artery? Yes, carotid artery, but we haven't learned the rest of that yet, so 1 mark just for not knowing anything. Oh, what the hell is this question? In the following passage. Oh, okay. An ecosystem supports a certain size of population of a species called the carrying capacity. Hold on, well, I've never seen a question like that before. It's the carrying capacity, because it can only have a certain amount. There are often numerous habitats within an ecosystem. Within a habitat, a species occupies a niche? governed by an adaptation to both abiotic and biotic. Why is this concerningly easy? Population of different species form a community, I want to say? Or an ecosystem. It's no, it's community. Yes, okay. Easy 2 marks. It is. I don't think there's anything you'd get here though, unfortunately. We are not talking about glucose. This is paper 2. Suggest 2 Suggest 2 reasons for conserving rainforests. Oh, I hate these questions because it makes you have to like state the obvious, which is really annoying because I don't think like that. 2 reasons for conservation. Oh my god, I can't think of any. Why would you want to conserve a rainforest? Okay, 2 wild ball answers. Something about protecting the organisms. Force leader of a group set mindset. Okay, hold on. Protecting organisms. Oh, I guess a better way of phrasing that would be like to prevent extinction of certain organisms. I swear there was a mark point. Yes! For preserving biodiversity. I was just going to say that. I swear there was a mark point when we did a question with Miss Lumley about preserving... Please say it right. Conserve/protect species/organisms. No! That would be a single mark point. Wow, okay, that's mean. Wow, that is a mean question. Conserve/protect species for biodiversity. That is 1 mark. That's nasty. Let's try and drill these other ones in, then. Conserve species for biodiversity. Source of medicine/drug/wood. Okay. I was, in the back of my head, I was thinking, would one be, it's a source of resources, but I wouldn't know how to phrase that. One is just for tourism. That's interesting. And reduces climate change. Okay. I could probably remember those. Give 3 reasons for the low efficiency of energy transfer from secondary... Oh, I hate this topic. Give 3 reasons for the low efficiency of energy transfer from secondary consumers to tertiary consumers. There's 3 reasons for that? Because obviously you have like your producers, then your primary, secondary, and then like the tertiary. True. 3 reasons for the low efficiency of energy transfer from secondary consumers to tertiary consumers. I'm gonna guess 1 is that the energy is lost. Oh, okay, I remember the mark scheme for this one. I swear I do. One of them is that not all of the animal can be eaten, or not all of organism can be eaten. Because like there's things like bones that most organisms can't eat. Lost through excretion, I'm gonna say. And also maybe like competition. Because if like, I don't know, this organism kills this one, but then like 20 others, different organisms eat it, then it's not passing the energy directly on. Let's see what it says. Okay, hold on. Excretion was one. Competition wasn't one. I don't think not all can be eaten would be one, which is weird because I've seen that in a mark scheme multiple times. This is the thing about biology, is the mark schemes every year change. So the answer one year might be incorrect another year. So the correct answer would have been heat loss from respiration, which I don't think we've ever gone over. Heat loss from respiration. I don't think we've ever gone over that. And also, food not digest. Oh yeah, I would have gotten a mark. Not all can be eaten. Okay. That was a mark point. Yeah. Yeah, it said not all eaten or food not digested, which I guess is fair. Either or. Man, these mark schemes are really tight. Okay. Please be some nice questions. Oh, have we gone over cancer yet? The gene codes for a protein. The protein relays signals from outside a cell to a nucleus. Suggest and explain how an alteration to the gene can cause a tumor to develop. Would this be something we haven't learned yet? I'm trying to recall. How an alteration in the gene can cause a tumor to develop. Oh, okay. I guess you could say number one, like mutation. Yeah, I could probably guess. Like, the thing is, I haven't actually gone over shit like this in a good while. What would it be specifically asking for? It's saying suggest, so how an alteration in the gene. So I'm gonna say, obviously, we'll just do a very quick example, like, if it's, for example, insertion mutation, where that obviously changed. I know one mark point is going to be different sequence of amino acid, which will lead to a different tertiary structure, or overall a different protein being produced. And I know that that'll then lead to this protein, like, hold on, I want to check to see what it... Yeah, okay, mutation changes the base sequence, which changes a protein. Result... Okay, yeah, I would have easily gotten that then, I think. Yeah. Accept mutation, or accept a named mutation. Accept change in amino acid sequence. I think that that's fair then, honestly. Different mutation. And then obviously the thing I was midway through writing there was going to be that it leads to uncontrolled mitosis. Any more questions? We have not done that yet, I don't think. DNA extraction, I don't think we've done that. Oh yeah, we have not done that. I don't know any of those words. Oh, okay, a question. A big essay. ATP is essential for muscle contraction. Describe the role of ATP in muscle contraction. We have literally just done this. This is the sliding filament hypothesis. You have your myosin. You have your actin. The myosin head has ADP on it, and what will happen is when the binding site is exposed because of the calcium ions released from the sarcoplasmic reticulum, this myosin head is going to bind to the actin. It is then going to change angles, which will pull the actin forwards. The ADP is then released. So the ADP is released, and ATP binds with the myosin head. This causes it to return to its original position. The energy from the hydrolysis of ATP is then used to cause it to change angles. So let me see. Why is it a hypothesis? Let's see what it says here. Yeah, to move the myosin heads so the actin filaments are moved inwards for active transport. Oh, okay, that is something thinking outside the box. Yeah, the calcium ions are obviously going to need to be moved from the sarcoplasmic reticulum to where we are here, the sarcoplasm. That's going to require ATP for active transport. I wouldn't have gotten any marks there though, because I was right. What I was talking about was right, but I didn't phrase any of it correctly. ATP will break the acto-myosin bridge. It will move the myosin head, so I would have gotten a mark. And that causes the filaments to be moved inwards. Okay. I think that's understandable then, because we have literally just done this today. It's obvious that I wouldn't be able to phrase it 100% right. But yeah, what I described basically, the ATP is unbinds from the head. The ATP attaches, which causes the head to return to its normal position, which breaks the cross bridge. And then it causes the head to obviously move. Okay. Nice. Describe and explain how taking creatine supplements and carbohydrate loading can improve performance of different types of muscle fibers. Oh, this is fast and slow twitch muscles. I don't remember shit about this, and we've just done it. This is like an essay question, though. Like, it has specifically written a big-ass essay about ATP here for you to read for information. Some studies have suggested that taking creatine supplements... What creatine is... Isn't that just a load of phosphate? Let me confirm before I start yapping. I swear it is. I swear that's what it is. Creatine. And I am getting a lot of adverts. What the fuck? Is creatine a lot of phosphates? Yes, because it's phosphocreatine. Okay. I could probably yap about this for 5 marks. What does it ask us for? Can improve performance of different types of muscle fibers. Okay, so some general mark points here. Creatine, that is obviously going to be used for the inorganic phosphate, for ADP plus PI, for energy, / ATP, which is obviously going to be used for the muscle contraction. I don't know how you would really word that though. That's just kind of some basic information about it. Carbohydrate loading will increase, I'm going to say, the glycogen supply, which is more glucose for aerobic / anaerobic. Primarily anaerobic, because it's the fast twitch muscle fibers which contain the anaerobic enzymes, obviously during glycolysis, which is quite a low amount of ATP being produced. So let's see what the mark scheme says. Creatine used to... Okay, I would have lost a mark just for saying It's not creatine. You have to say that it is used to form. To be fair, we have not gone over this very much, so I did not know that. I thought creatine was just the base. Phosphocreatine combines with ADP to form ATP. Nice. So 1 mark. Carbohydrates stored as glycogen. 1 mark. Glycogen hydrolyzed to glucose. I might have put that in an exam. Thing is, when I'm doing these streams, I'm writing and thinking incredibly fast. Because, like, time is of the essence. In an exam, I would take my time. And I would literally, in an exam, if this was the page, if this was the question and this was the space we had, I would make sure every line had a new bit of information on. Like, I would fill the entire page. Here, I'm adding, like, 1 note max. So, I'm not coping. I probably would have put that in an exam. Glycogen hydrolyzed to glucose, yeah. Glucose used for respiration. Really? No marks for talking about anaerobic? Accept glycolysis. Okay, yeah. So I would have gotten 3 out of 5 there, I think. I mentioned glycogen storage. I mentioned combining with PI, the ADP, to form ATP. I mentioned glycolysis. The 2 easy things I could have mentioned is this. It's used to form phosphocreatine. That would have got me up to 4 marks. And then I could have easily mentioned the hydrolysis of glycogen for 5. So, okay. I'm fairly happy with this. I'm fairly happy. I'm probably going to have to go in like 5-10 minutes. So, are there any other questions? Or is that the whole paper? Suggest how transport proteins are beneficial during exercise. Oh, come on. They're glucose transport proteins and fatty acid transport proteins. Okay, we could probably milk 1 or 2 marks out of this, I want to say. Fatty acids, that would be for lipids. I'm trying to think what the relevance of lipids would be in muscle contraction. Wait, CD36. Oh, hold on. Wait. During aerobic, calcium ions have several roles. They activate the inclusion of glucose transport proteins, and the inclusion of fatty acid transport proteins. Okay. So it's basically just asking us what is the purpose of glucose and fatty acids. I guess you could say then, how are they beneficial during exercise? Because I guess a general marking point would be that they allow for fast, facilitated diffusion of glucose so that it's readily available for respiration. Fatty acids, I don't recall triglycerides. I'm trying to recall the relevance of fatty acids. This is going to be a really obvious mark point when I check the mark scheme, isn't it? I'm going to hit myself in the head with how obvious it is. We have not learned about the relevance of fatty acids during muscle contraction. Oh, during exercise though. Whenever it mentions exercise, you have to milk the fact that it's for respiration. Fatty acids during exercise. I don't recall. Okay, yeah, that is... Okay. I'm guessing that probably is what it is. We don't do much about lipids, to be honest. Not since year 12 anyways. More glucose entering the muscle cells by facilitated diffusion. Fatty acids used in Krebs cycle. So that's the only mark you could have got. Are they used in the Krebs cycle? Oh my god, I faintly remember that. But oh wow. That is kind of mad. Respiration provides more ATP. 2 marks. I think that would be 2 marks. Because I said for respiration, I didn't mention ATP. Okay. I'm happy then. That would be 2 out. Wait, how many marks was his total? 3. I'm happy with that. A 20 marker on fatty acids. Bro, that would kill me. That would actually kill me. An increase in muscle activity causes an increase in heart rate. I love this. No, I love this. This is the feedback loop, right? An increase in muscle activity is going to be an increase in respiration. That is going to be an increase in a decrease of the pH level in the bloodstream because CO2. Okay. So that is going to be detected by chemoreceptors in the carotid artery. That is then going to be sent by the parasympathetic? No, sympathetic pathway to the medulla, which is going to increase the frequency of nerve impulses to the sinoatrial node, which will cause an increase in atrial contractions, increasing the heart rate. What the yap? Increased CO2 detected by chemoreceptors. Send more impulses to cardiac sensor. More impulses along the sympathetic pathway to sinoatrial node. Easy 4 marks. Nice, okay. And that's it. That's the 2023 paper 2 done. Obviously, I couldn't answer everything there because we are still about... I think we're about 2/3 done now with the year 13 content. But I am really happy with that, I think. I'm really happy with that. There were some tough questions though. The ones we did a couple of days ago in particular were very difficult, but I'm more than happy to call it quits there. I'm more than happy to call it quits there. Thank you guys for watching, obviously. I appreciate it, Ethan. Man, I need to go take a sit down now. And yeah, there are probably 6 more papers for paper 2. Feel like we could get 1 a week done. And by that logic, we should be done soon.